second. I think, yeah, I think we're live.
Well, welcome everyone from Canada all the way down to New Zealand, because I know that we have viewers down there as well. This is the second in our series of conversations about COVID-19 and more from the McGill Office for Science and Society. And let me just give you a very quick rundown on who we are and what we do. Our office was established about 20 years ago, and our task really is to separate sense from nonsense and to separate fact from fiction. We try to make sure that you're all up to date on what is happening in the world of science. And hopefully, if all goes well, we also try to keep you out of the clutches of charlatans. And of course, they are emerging in droves these days in response to the fact that science doesn't have all of the answers to COVID-19. Let me just introduce you to who we have online here. We have Jonathan Jarry, who's a science communicator in our office, background molecular biology. We have Dr. Debbie Schwarcz, and she spells her name the same way as I do, and there's good reason for that. And she's an emergency room physician, and she'll bring us up to date on what is happening in the hospital. Unfortunately, our colleague Ada McVean, who was with us last week, isn't with us, and there's a good reason for that. She has tested positive for COVID-19 and is not feeling great, and neither is her partner Matteo, but they're getting better. And they were pretty run down, and they got better, and then it struck back. And this is a pattern that, unfortunately, many people are reporting. So we'll start out with that, Debbie. Maybe you can comment on that. Sometimes people think that they're over the hump, and then it comes back with a vengeance.
Yeah, so that's exactly the case. And I think what's important to talk about here is the fact that even there are some people who are sick who are actually getting false negatives. So it's this false sense of reassurance when you get your test back, because it doesn't necessarily mean that you're free of the disease. So that's definitely very concerning. And then exactly what you said, that we're tending to see a little bit of improvement, and you feel that you're over that hump, and then unfortunately another episode of worsening. But we don't know why.
About when we should be peaking. What are you seeing in your hospital in terms of number of patients who are presenting?
We're starting to go up. We're definitely climbing that curve. Patients are coming in more and more now. In the beginning, it was definitely, I know when we spoke last week, I talked about how quiet it is in the emergency room. And while it's still quiet in a sense, with regards to our usual emergency population, now what is really starting to ramp up is these patients that are coming in with symptoms of COVID. So we're definitely seeing an increase now. I think that the peak is being projected at this point. The peak is being projected at this point to be in approximately 1 week from now. So we should be seeing by the end of next week is when the projected peak is. But bear in mind that this peak we haven't been on the ball with getting it, unfortunately, because it's very hard to predict these things.
Well, of course, there's a tremendous amount of research on trying to find something that helps. And Jonathan, I know that you've been keeping up to date on what's happening with the plasma research and immunity. So give us a little bit of background there.
Sure. So there is this idea that's been floating around of, when you recover from the disease, you will have antibodies in your blood, which are these little Y-shaped molecules that will recognize the invader, in this case, the coronavirus. And so what if you could take those antibodies that you've made and put them in the body of somebody who is struggling with the infection to sort of boost their defenses? And it's a very old idea. In fact, it's something that was used fairly routinely in the late 1800s or early 1900s, before we had antibiotics, before we had vaccines, before we had a lot of drugs. And whenever there's been an outbreak of a particular type of disease where the situation is very urgent, For example, the Ebola virus, it has been tried, this idea of taking part of a survivor's blood and taking a fraction from it, that it's usually the plasma, which is the sort of clear yellow liquid that is full of antibodies, and infusing that into somebody's body. Unfortunately, whenever it has been tried in recent times, the studies have been very, very poorly done, in large part, I suspect, because you're sort of desperate and doctors are just trying to save lives. So we don't really know if it works for these kinds of diseases. But now it's very interesting to see that there's a large Canadian clinical trial that is about to happen to test whether or not this could work for COVID-19. So I think about 40 hospitals throughout the country have been recruited to very rigorously test that hypothesis. Can we take the blood of somebody who has survived COVID-19, who has made antibodies against it, and give it to somebody who is struggling with the disease? Will that make a difference on their recovery, on whether or not they will die, all of those kinds of things? So that's something to look forward to. It's going to take a few months before we have results, but at least it seems to be studied in a rigorous way, as opposed to a lot of the past studies that have come out that have been kind of not nearly as rigorous.
This is very important to emphasize that the way that science works is through proper controlled trials, and not through hunches, and not through releasing information before it has been peer reviewed. The reason I mentioned that, because I'm sure that many of you have seen yesterday or today a supposed Belgian study that shows that if you're out walking or if you're out bicycling, you have to be extra special careful. In fact, keep 60 to 70 feet back from somebody because they're spewing viruses. Very important to understand that there is no such study. What they are talking about is a simulation that was published. Well, actually, it wasn't even published. It was a newspaper article. So there isn't even a study that we can talk about. There's nothing to critique. All it is a newspaper article. A reporter talked to a researcher who did this simulation on a computer. So we really can't say anything about this. And any suggestion that exercising outdoors is counterproductive, I think at this stage is very unfair, and certainly putting the cart before the horse, as this newspaper article suggests. And it's going to scare people from walking outside. And if there is something to this, let him publish it. And let the scientific community take a look to see, what is really going on here. But this is spreading around the internet like wildfire, with no documented evidence whatsoever.
But one thing that I think people do need to be aware of, that I've also been reading and experiencing because I am a runner, is that the nicer the weather gets, it becomes increasingly challenged to social distance, and so you kind of end up with a runner and a walker and deciding who's going where. Am I going on the road? Am I going on the sidewalk? And I know that in my experience, when I go along the Lachine Canal here on a nice day, I can tell who are partners or like they're socially distancing together because they're walking side by side, and others who are friends who are going for a walk, but there's a big space in between them. And as a runner, that makes it very difficult because I then have to go between them. And at that point, I'm not keeping social distance. So I almost sometimes want to yell.
If only you had a stationary bike indoors.
Oh, yeah, which also I do have, yeah. But it ends up being this, it is going to become more challenging as the weather gets nicer to navigate that. But I do think for one's mental health and health in general, to still keep active. But that was something I was thinking about today.
There's so much nonsense out there. There's this circulating document supposedly from Stanford. It does not come from Stanford, which tells people that if they can hold their breath for 10 seconds and not cough, it means that they don't have the virus. That'd be what
That's what I think about it.
Yeah, it's totally ridiculous. But something that is not ridiculous is the change in the advisory about when to start intubating someone.
Yes. Yeah. There's more and more evidence now coming out. I think obviously we're learning from other countries. We're learning from the people who have already gone through this, from what they did in Italy, what they're doing in New York right now. And whereas initially our protocols were early intubation.
Can you explain what an intubation is?
Yeah, sure. So intubation is using it's a plastic tube, and we pass it through the vocal cords so that we could attach the tube to a ventilator, a machine, to breathe for the patient. So a lot of times the patients who have COVID are in acute respiratory distress. So they come in and they're kind of they're so tired they're so tired because they've been working so hard to breathe that they actually can't even breathe anymore, believe it or not. So that at that point, obviously, I mean, we would have no choice but to intubate. But there are some procedures that can be done before that at first we were very much shying away from because of the risk of aerosolization. So these are called non-invasive ventilation techniques, which would include CPAP and also another type of technique called high flow oxygenation. So which is basically like, it looks like nasal prongs that go inside of the nose, but it's sort of closed around the nose. So we think that there's less aerosolization with that. And the reason why we're leaning away from intubating, if we can, obviously, is because patients are not doing well on the vents. And we don't really know why. We don't know if it's we're not treating them properly. I mean, I can't talk too much about that because, like, I work in the emergency room. I'm not an ICU doctor, so I'm obviously not managing the vent settings. But I know that initially there was a lot of understanding that the virus caused kind of like an acute respiratory distress syndrome in the patients, which is treated a certain way with very high pressures being given with the oxygen through the ventilator. But now we're thinking that those high pressures are actually what is being what is causing lung damage for these patients. So, this is also new and it's all changing so quickly. So it's very hard to know what to do. And we're just kind of trying to stay on top of everything and stay on top of all the, it's not even at this point research, because we're just following what people are doing, and if it works or if it doesn't work, because this is all just changing daily.
There's another theory that's starting to circulate, and that is that the virus is actually doing its damage by destroying porphyrin molecules, which are part of hemoglobin, and that the reason that there is all this damage to the lungs is secondary to the fact that they're not getting enough oxygen because the hemoglobin is being destroyed. Now, of course, this is just a theory that is floating around, but there are people who are paying attention to this. I mean, the fact is there's so much here that we don't know.
Yeah. There's another theory. You had just sent me the YouTube video of it, but I had already seen it, of a doctor in New York who is thinking that this is actually similar to high altitude sickness. Yeah. So I think that's the scary part about this, is that we just don't know. This is not something that we have textbooks about to help us. So we're kind of figuring things out on the fly, unfortunately.
Of course, there's plenty of real stuff to be scared about, but there are also some interesting questions about sort of minor things that come up. Over the last couple of days, I had several people ask me if it's okay to give flowers as presents to someone, and could this transmit the virus? Well, one thing that we have learned in this business is that you cannot say impossible about anything, because you can conjure up a theoretical scenario where someone would have coughed or sneezed on those flowers and they're delivered to someone and they touch the flowers and then they touch their face. It's a possibility. I think it's a very unlikely kind of scenario, but it kind of shows what people are worried about. And they're worried about absolutely everything. And, you're out walking on the street, you look at everyone who's walking out there as if they were a walking time bomb. And that, you have to stay away from them. And it's amazing how this has just changed the world. Yeah.
No, I was just going to say that we, I just have a question here on Facebook that I was looking at, that this switches us again just back to UV light, which we also spoke about last week, and killing the virus. And
Right.
I have a feeling that this question is going to say that, I mean, no one knows, but how effective, again, we're talking about runners and bikers, and
It's
how
UV light
effective,
is extremely effective.
but not only,
It's
but UV light,
but
but then
right.
talking about sun. How effective is sun
Yeah.
at killing or inactivating the virus when you're talking about runner A going to runner B and you're outside?
Here's the story with UV light. UV light, of course, is a spectrum of wavelengths. And the very short wavelength UV light is very effective at destroying the virus. The longer wavelengths are not quite as effective. And UVC, UVA, and UVB all come from the sun. But the ozone layer protects us from too much UVC, which can be highly damaging. Not only does it damage viruses and bacteria, it damages human tissues as well. So not much UVC comes through from the sun. A little bit does. So anyway, outdoors is a good chance that the virus is going to be inactivated. And of course, with time, the virus does get inactivated. I just wrote a piece that we have on our website now about what happens, what is this inactivation? Because I think that's something that people really haven't addressed. You know, you talk about that on paper, it lives for 24 hours, on plastic or steel, it lives for 2-3 days. And what happens? Because a virus isn't really living in the sense that it reproduces. It can't reproduce by itself. So what do we really mean when a virus, quote, dies? So I try to explain that. It's a bit complex, but it's on our website, and it will be in our newsletter on Saturday. Okay, any other questions that come up on Facebook?
Very quickly about UVC, it's also a question of time. For example, I have this device that uses UVC to disinfect my phone. It takes 10 minutes of constant irradiation to kill all the microorganisms. So just the fact that we have sunlight that has ultraviolet light in it, and that will shine on an excretion from a runner for about 5 seconds or 2 seconds before it hits you, it wouldn't be enough in this particular scenario.
That's not that's not really what is being asked mostly by people. It's if you put some object outdoors that they think.
It's what was being asked on the stream, actually.
Yeah. The other thing with UV light, you have to remember that it falls off with the square of the distance. So if you have one of these UV lamps that are sold, disinfect counters, etc., it has to be very close to the surface that you're disinfecting. You can't just flash it around the room and think that you're going to disinfect the room. That is not going to happen.
So we have a question. So those UV devices, Jonathan, I think we spoke about this last week also. Someone wants to know where you get them, but I think that they're sold out, as we said.
I think they are sold out, yeah. I mean, I'm not doing publicity for the company. Mine is from Phone Soap, one word, but I've heard from a friend that they're sold out now. So,
actually, we have a comment here on YouTube. There's a ton of Facebook posts that suggest that the cause of the virus is 5G.
Right.
Care to share some info how that could possibly even
ridiculous thing that is barely worth addressing. It is so absurd. 5G, the G stands for generation. This is 5th generation technology, Wi-Fi technology, and there's absolutely no relationship that has ever been shown between this and the virus. But these conspiracy theories are par for the course whenever you have a situation for which we really don't have answers. Another thing, many people may have seen distributed today this supposed article from the BBC that Sophie Gregoire, the Prime Minister's wife, and Tom Hanks were using a Swiss formulation in order to treat themselves. And it looks like a legitimate BBC website, which it isn't. This is a scam website, and it is hyping a product which is glutathione. Oral glutathione is not effective against anything because it is immediately digested, but it looks authentic. And you read this, and it talks about Swiss researchers, and they're doing this in secret, and only if you're someone like the Prime Minister's wife or an actor, you have access to this. But now the company is making it available to everyone. So this is just total nonsense. But you have to be very careful with this because these websites look very authentic. Also, Jonathan, maybe you want to comment on robocalls, which are, supposedly from the World Health Organization or other organizations asking for your credit card so that they can send you a test kit or something like that.
I see. I hadn't heard of those, but yeah, it's a fairly commonly used way of getting your information and selling you nonsense. The WHO is not going to send you an automated call and ask for your credit card number. I think that's a good rule of thumb to keep in mind.
Yeah, the thing to do is hang up. Okay, something we talked about time, and we're going to talk about this a lot more, is hydroxychloroquine. And of course, this is being pushed in what I think is a totally ridiculous fashion by President Trump. But they are using it, and there are actually controlled trials that are underway. So we will see. So Debbie, what is happening in the hospital now with hydroxychloroquine?
Emily, did you want to say something?
With the hydroxychloroquine, but also maybe then you could go into we have a bunch of questions on ivermectin, if I'm saying it right.
Yeah.
Yeah, we have a lot.
But let's talk first with the hydroxychloroquine.
So the hydroxychloroquine, there's actually a big McGill trial on this now. It's specifically for healthcare workers, though. So there is research being done on it, and it is in our protocol right now for patients who are admitted in the ICU.
So this is the kind of thing that is done basically out of desperation when there's nothing else. And there's some in vitro, meaning in laboratory evidence that it may do something. But we really need the proper controlled trials, which luckily are underway. And this is the same thing with ivermectin. This is an antiparasitic drug. It has a long history of use, but it's never been used in a virus infection. But again, there's some laboratory evidence that it may block the action of the virus. But it needs to undergo controlled trials before starting to jump on the bandwagon, because it's not only a question of the side effect profile. For example, with hydroxychloroquine, there's pretty good knowledge about the side effect profile. I mean, you can get various kinds of cardiac arrhythmias, and there are complications. But what really is not known is how it can interact with this condition, the virus, because a drug that has been approved for some condition and has been tested, the side effects apply to that condition and the use of that drug in that condition. Here, no one has yet tried hydroxychloroquine for this viral infection, and it may have a totally different side effect profile. Very possible, because already in Sweden, where they were using it in some hospitals, they have stopped using it. In France also, where that first provocative study came out, they have now stopped using it because they have started to see side effects. So I think we really have to wait until the controlled trials come out.
They also actually mentioned just your thoughts on the Bacillus Calmette-Guerin vaccine.
So I just wrote about this. It's going to be published on Saturday. Again, we're at a point where it's a little bit like you're buying a bunch of lottery tickets to see, knowing full well that most of them will be duds, but hopefully that one of them will change your life. The BCG vaccine is a vaccine that was developed to fight against tuberculosis. It was tested in the 1920s. And what's happened is that as the prevalence, how common tuberculosis is, as it has gone down in a lot of developed countries, fewer and fewer children are mandated to be vaccinated using this particular vaccine. And so there are countries where very few people have received the vaccine, and there are countries where everybody gets vaccinated with this BCG vaccine. What's happened is that there's a manuscript out there that came out just a few days ago, maybe a week ago, by a professor in New York. He's an electrical engineer who studies the human brain, and he and a bunch of students got together and they looked at the data, and I use the term loosely published, but they put a manuscript out there on a preprint server, meaning that this was not accepted by a scientific journal yet. It hasn't undergone peer review yet, so no other scientists have looked at it before it was published. It's a little bit like publishing a blog post on a website that is read by a lot of scientists. And what they have shown is an association. Given the data that they extracted all over the world, they are claiming that in countries where BCG vaccination against tuberculosis is universal, everybody gets it, the number of people who get COVID-19 in that country and the number of deaths that are related to COVID-19 are seemingly much lower than in countries where we're not using the vaccine universally. So what they are claiming is that, well, it's possible that this BCG vaccine might protect you against COVID-19. Now, this is quite a leap, and there have been a lot of criticisms of this particular manuscript. But again, because we are desperate and because there is some evidence that this particular vaccine might actually give you broader immunity, it might help your immune system to fight off other infections, not just tuberculosis. It's limited evidence, but there is some evidence. There are some countries that have started clinical trials of this vaccine amongst their healthcare workers. So some of their workers are getting the vaccine and others are getting a placebo to see if it might help. So the data is not great at the moment, but again, it is one more thing that is being investigated right now.
But Jonathan, maybe you want to explain also, since you're on this topic, that someone said that they have a postdoc currently working in the lab on biophysical testing of potential inhibitors of the main protease of the SARS-CoV-2 virus. What does that mean to a non-scientist?
Biophysical inhibitors of the main protease. Okay. Well, the virus itself is it's like a little organism. It's not quite alive by our traditional standards, but what it is, it has a blueprint inside that is made of RNA. And it tells our cells how to make more copies of this virus. And the virus itself has an array of proteins, which are like the effectors, the things that are doing the work and that help with the structure of the virus. And also has a lipid coating, which is a fat envelope. And so there are different units of this virus, different molecules in the virus on which we can act if we have the right drug, for example, right? So if you can target a specific molecule on that virus with one of your own molecules, like a drug, for example, then that's one way of deactivating this virus and of helping people who are going through this infection. So what this postdoc is doing, what a lot of other people are doing, is they're trying to figure out what is the Achilles heel of this virus, which protein of it can I act upon with another molecule in order to, quote unquote, kill this virus.
There's another interesting question that was asked several times, surprisingly, during the last week. With mosquito season coming and black flies coming, what is the chance that if one of these insects lands on my face, and let's say I'm infected, and then it lands on someone else's face, and then they kind of touch their face, smearing the potential virus on there. What is the chance of acquiring infection like this? What can you say? Again, it's a theoretical possibility. You just can't rule anything out. But I think this is so unlikely that this should really be way down on our list of worries. The flies, the mosquitoes are not going to be a major way that this virus is transmitted.
Well, with every new season, we're going to get new questions. I know that in my group of friends, we were talking about pools and like having people over and swimming. And is chlorine going to kill the virus?
Chlorine kills the virus. There's no question about that. You don't have to worry about swimming in the pool. What you have to worry about is in the changing room, where you're going to be next to people who are changing. Talking about changing rooms, Debbie, this is something that we talked about yesterday. Is that in hospitals where there are dedicated rooms where physicians take off the PPEs, the protective apparel, and that is one place in the hospital where they're actually finding aerosols in the air, because when you take off the gowns, they crinkle and everything, and the virus is aerosolized. Have you?
Well, that's why when there's proper procedures for both the donning and the doffing, and you really have to be very specific about it. And we've had training sessions about this, and you're always supposed to have somebody watching you to make sure that you're doing it properly, because exactly that's the doffing is the period of time where you're most at risk of getting contaminated. It's not actually during the period when you're examining the patient, it's afterwards. So that's definitely high risk. And that kind of brings back the masks, everybody. So now actually in the hospital, we've been mandated to wear surgical masks all of the time, by the way. Because we had talked about that last week, about whether or not we should be wearing masks. And so that has been something that has changed. And I think that I mean, I think that it's a good idea because the healthcare workers are prime vectors for coronavirus. It's, I think if we're in the healthcare setting, it's the nurse. You should see how close the nurses and the doctors and the maintenance and the RTs. I mean, we're all working so close together. It's very hard to practice social distancing in the hospital.
Can we
Talking about all protective equipment, do you have enough?
No. No, so I think that we've all been reassured that we will not be, we will not be put in the position where we will have to go and see a patient without the proper protective equipment. Right now we have enough, but there definitely are shortages. So that is a little bit scary for us. Feeling like we don't have the proper protection. And even as it is, the protection that we do have is, I mean, it's pretty measly when you compare it to the protection that you see them wearing in New York and Israel. We're not wearing the full overall suit with the hoods. We don't have that. So we're all feeling a little bit vulnerable. But we're trying to take it day by day, and I have to get fitted for another type of mask now to see if if the N95, another N95 mask, because the one that I wear is now is one of the ones that's in shortage.
And also the N95 masks, they're different models, and you have to wear the one that fits your face.
Well, that's what I'm talking about. So I have one that I have been fitted for, and I have to go get a fitting now for another one because the one that I am fitted for is currently a shortage of it. So that's why you see people going out wearing N95 masks, and it's beyond silly. If even just for the fact that it's not fitting them properly, and if you're touching your mask, you're contaminating yourself.
Well, someone's asking, are you re-sterilizing your N95? And,
yes.
And when do you use them?
We only use them for aerosolizing generating procedures. So that would be things like intubation, extubation, bronchoscopies. Non-invasive ventilation is another one. And I saw somebody had asked previously about CPAP, about their partner being on CPAP and they're infected. And yes, that does put you at risk. So you shouldn't be sharing the bedroom with that partner anyway, if he's positive or she's positive. But yes, the CPAP is putting you even at greater risk. So we are starting to use them more than once. We are not re-sterilizing them right now because it seems that the heating them takes away from their benefits. So the elastics don't work as well. So ultimately, they don't fit your face as well. So what we're doing right now, I mean, it sounds kind of funny to be completely honest, but when they come off, as long as you haven't been the one to, so for me, primarily, I'm going to be using it in a resuscitation case. So if somebody is requiring intubation. So if I'm the one that intubates the patient, it goes in the garbage. But if I'm the team leader, so I'm not the one who's intubating the patient, then I can keep it. And you take it off and you're making sure not to contaminate it. And we're putting it into a paper bag with our name on it, leaving it for 3 days because the virus is going to be inactivated after 3 days. And we have it on the sideline if ever we should need it. So we are considering the possibility of having to reuse it, but we're not quite there yet.
There's really vigorous research everywhere now on what to do, how to reuse masks, and trying all kinds of techniques, ethylene oxide sterilization, hydrogen peroxide vapor, which seems to work well. That doesn't destroy the mask. They're using UV light, using heat. So yeah, I mean, there will be some procedures found that work. But also the industry, of course, is gearing up on producing more. And I just heard that Canada is receiving 10 million N95 masks from China.
Well, that leads, someone said here on Facebook, the cloth masks that people are making at home, whether hospitals have any use for those?
No. No, I mean, I think that if you want to go out in public, that would be the place to use those. I think that, those are going to protect you against, I mean, mostly gross respiratory droplets, but I would definitely not feel comfortable wearing that myself in the emergency room. One of my colleagues had joked about like creating a UV box that you just kind of step into and decontaminate yourself.
Yeah, well, I don't think you want to step into a UVC box.
Yeah. Just put lots of sunscreen on.
Yeah. Hold on. Let's go. We have a few good ones here. We started off the stream talking about our colleague Ada and her partner who had the tests, and then there was that period of time, and they ended up day 11 just having his worse symptoms even at the beginning. So someone's asking about the 14-day isolation that's required for travelers and even, quarantine. How did the 14 days get determined? I mean, how did this happen? If someone doesn't come into contact with anyone during the 14 days, are they COVID negative after? I mean, what happens?
The incubation period is 10 to 14 days. So that's where the 14 days came from. So the idea is that if you have not exhibited any symptoms at all within those 14 days, then you don't have COVID. Now, the problem is that once you've been quarantined for 2 weeks, and if you decide to go out and you go to the grocery store, you can contract the virus there. So, even if you're not seeing anybody, I mean, if you step outside, as we talked about, when you're running outside, there is a risk there.
And now, of course, there's the whole issue of asymptomatic people.
Yes.
Right. I mean, you can be a carrier and be perfectly fine, and so that's also something that needs to be researched more. But virtually every day we're learning something about this. Prince Charles, why do we bring him up? Because of the story that came out today from India, and actually from the minister of a government organization in India that looks at alternative medicine, Ayurvedic medicine. They actually regulate this thing. And he is claiming that Prince Charles got better very quickly after he tested positive because he was using a homeopathic remedy. And India, of course, is very big on homeopathy. And this is the minister who is claiming that Prince Charles took a homeopathic remedy and that's why he got better. And of course, they're using this now as advisory for using homeopathic stuff. Now, the prince's office has denied this. And it probably is true that he didn't use this. I think he's smart enough not to do this, although he is a believer in homeopathy. But I think when it comes to his own health, I think he probably will have second thoughts about homeopathy, which is the most absurd of all the alternative medical practices that are out there.
So with some absurd treatments, I mean, someone's asking...
Oh, Emily, you're on mute.
Yeah, we can't hear you.
But I was going to answer one of the questions here because somebody asked about the false negative rate, and I did allude to that initially. There's a very significant false negative rate. I can speak for patients that I'm seeing in the emergency room right now. We're repeating the test. So first of all, I'm getting some patients that I'm certain that they have COVID just based on their presentation, their chest X-ray findings, their oxygen saturation levels. Just from the short experience that I have with COVID, and then their test comes back negative, which I find I don't buy it. And then there's other patients that are testing negative, and then we're repeating it 2 or 3 days later, and it comes back positive. And there have even been cases that have been multiple negatives coming back finally with a positive. So basically, I'm not too reassured by negatives at this point. I'm reassured by positives that they do have COVID, but I'm not finding the negatives reassuring. And it's I think that time will tell what we're going to be doing in these situations, because as it is right now, we're pulling off the precautions once the test comes back negative. But I feel like it's a little bit too early.
Well, when they swab, I understand that you can swab either the back of the throat, right?
Yeah.
Swab through the nose.
So that's another problem, I think, is that it's quite operator dependent. There is some area for error, because depending on how far the swab goes, the swab should go extremely far back. Like, this is a test that, unfortunately, I've had it done myself, and unfortunately, it is quite painful. I would say it's not uncomfortable. It's painful.
It goes close to your ear, right?
It feels like it goes into your brain. But no, it's quite painful, actually. And interestingly, I've had 2 done within the past couple of months, because I had I had influenza a few months ago, or a month or 2 ago, and that one was extremely painful. And then interestingly, because I mentioned that I had a sore throat, I think it was 2 weeks ago now, and I was swabbed for corona, and it was not nearly as far back, and it was negative, but I am better. So I think that there's a lot of, it does need to go very far back. And so you do need people that are properly trained to do it.
Well, the tests are going to get better. And now supposedly with the Abbott test, which can be done from 15 to 30 minutes, or as President Trump says, in 5 minutes, which of course is not true. And this is being spread around now in the US, but they need the special machine into which you put the sample. And apparently, although they've been sent around, they're not being used. No one can explain exactly what is happening there. So there's a lot of talk, but so far that test is not being applied. But we will have proper testing, I think. And that's going to be the key. Because really the only way that you can get a handle on how this thing is spreading, which is, so mysterious in many ways, is to keep testing and see if someone tested positive, and then you test them and they become negative. Then you test them again, become negative, test them again to make sure that, you don't become positive again. That's the only way that we will eventually know how this thing is spreading. Because at this point, it's still possible that someone doesn't develop immediate immunity to this, and that you can still spread it even when you think that you're not spreading it.
Well, how long after the 14 days? I mean, after day 14, if you're feeling totally fine, you're okay to just go out and resume normal life?
There is no more normal life.
No, that's true. But
are you talking about if you've been diagnosed?
Yes, if you've been diagnosed, and then the question.
No, I would not. So if for healthcare workers, actually, we have to be retested and have 2 negatives within a period of 24 hours afterwards, in order to return to work. And I know of a colleague who had tested positive, and 3 weeks later, who's completely asymptomatic, and still has, she's had 2 positives within the past 24 hours. So it becomes very frustrating, because you're kind of stuck on the sidelines at this point, and you want to get back and help, because, everyone feels so helpless. And, this is the one time where I think we all we want to go into work. It's not to mention it's a break from the kids.
I think we're coming to the end of our time. And the questions, of course, I'm sure there's many there that we've not been able to answer. There's many points that we haven't been able to discuss, but of course we will do this again next week, by which time things will have changed and new information will have come out. Some of the stuff we talked about now probably will be no longer correct. But one issue that we do have to keep an eye on is this business of fecal-oral transfer, because we've had this past week a number of studies, mostly from China, showing that there is the possibility of such transfer, because the virus does show up in fecal matter, in some cases even in the urine. Now, one thing that we have to mention is that when they do all of this testing, they don't actually test for viable virus. They test for RNA fragments. That is, the virus was present there at some time, still may be there, but it's not a test for, quote, live virus. And there's concern that because it's present in fecal matter, that when you flush a toilet, it gets aerosolized. So you should always close the toilet lid whenever you're flushing. I mean, that's a good idea also. But now it is showing up in sewage as well. Now, that has raised a lot of issues, because if it shows up in sewage, which it has, at least RNA fragments of the virus have shown up, the question is, does our water purification technique take care of this? And I think here we can give a pretty definitive answer. The answer is yes, because whether you're talking about chlorination, whether you're talking about ozonation, this virus cannot survive that. So I don't think that there's an issue with drinking water, even though the virus may be present in sewage. So I think there we can relax. You don't have to worry about your tap water having the virus in it. So I guess that's about it. And of course, you can always email your questions, and you can email them to me directly, joe.schwartz@mcgill.ca.
And any
or you can email them.
Any questions?
Emily, where else can they email?
Well, they can email, like you said, Joe Schwartz, and same with Jonathan Jarry and Debbie Schwartz. All our emails are on our webpage. We also have a weekly newsletter that you should sign up for, and it has all COVID-19 coverage and other. And any questions like last week that we did not get to, we will look on our Facebook page and answer those. And we hope to see you next week, next Thursday again. Stay tuned.
And like I always say, I hope until we meet again next week, I hope all the chemistry in your life comes out just right.
Thanks, everyone. Bye.